Those are the experiments that is from life, or this was called as the theory of biogenesis or abiogenesis previously. Okay, and there are certain movements like diffusion. No, we just did a little concept in which we studied that the cell theory, there is a third part that these new cells arise from the pre-existing one. Okay, so this point is basically based on the Reddy's experiment and the Pasteur experiment. So okay, and then the other new thing is the movement, the diffusion and osmosis. Though I'll explain you in a really easy form. There's no need to, you know them already. Okay, those are really easy. And what else is new? We have done all the type of microscopes already. Uh, Sorry. And then we discussed the stem cells in detail. We did those as well. So there is not much new in this, so let's start from the very beginning. So the okay, sorry, I was supposed to share the screen. Just give me a minute. Oh, it's shared. It's shared, right? Okay. So let's start from the cell theory. So later on we'll move on to the next. Okay, in cell theory we discuss three parts. There are three postulates, let's say, of cell theory. Number one is the cell is the basic unit of life. What is the second point? Third, I have already to, sorry, J. That is the third postulate. What is the second one? All living organisms are composed up of cells. All living organisms are composed of cells, and new cells arise from pre-existing cell. Okay, let's discuss this third point. Okay, since whenever a theory or a postulate needs or a principle, okay, there is a scientific process, the whole scientific process in which there are like certain categories. Let's say scientific method. We follow few steps. The very first step is we observe something, then we made a hypothesis about this. Hypothesis mean a rough idea of what might happen. Okay, like if we can see colors, why can we see colors? Let's say because we have some receptors for light in our eye. That is our hypothesis. Okay, and then experiments are made to prove the hypothesis. Okay, these experiments may prove your hypothesis correct or may prove it wrong. Okay, okay, then based on the results of the hypothesis, we can name them as theory or principle or law. You might have heard of the name law, like Charles law, Boyle law. In chemistry it's very common. Okay, so okay, what is the difference between theory and the principle? Okay, let's say if you have performed some experiment and it's and your experiments are proving your hypothesis correct again and again, then it would become theory. Okay, and if this theory is worldwide accepted by the by all the scientists, by and they also prove the same thing with the experiments, with different sort of experiments, then this that theory would become a law or a principle. Okay, this is the general thing that scientists follow. Okay, there was one of the disease that we discuss usually. That one is malaria. So it was observed that the people who lived in marshy places uh, have got more malaria. This is the observation. So many people believe the hypothesis was that might be drinking those marshy water can cause malaria. But later on, experiment proved that it's not marshy place or the marshy water. It's the mosquito that is present over there. And by repeated experiments, there was a theory that the plasmodium is the cause of malaria. Okay, this is the general thing that we like. I've given you one example. Let's apply this on this third postulate. So there were two scientists who design an experiment. The very first one is the Reddies. What did okay he's an Italian scientist basically, and he said that he observed that a rotten piece of meat was lying, and after few days the maggots they start to appear from the or they start to produce from those that rotten meat. There was a rotten meat, and few days later there were maggots. So to prove his point, whether because meat is rotten it's dead, so how can dead thing give arise to a living thing which is maggot? So he did an experiment. He did two different experiment. Number one experiment was the control experiment. Okay, he took a meat, place it in a jar, and then he allow house fly to sit on that meat, and after few days he observed, what was the observation, that maggots produced from that rotten meat. Okay, okay. While in the other experiment, now he covered the jar, uh, okay, covered the jar with meat. Now flies cannot enter, right? So when flies cannot enter, so no maggots produce. Okay, this was the observation. It's a very simple experiment. I think so you have got this experiment. Okay. Oh, okay. Okay, you want to learn that one as well. Okay. So there are three to four like terms that we use. One is the control group. Control group is the one in which you provide all the material except one thing that you want to prove. For example, if you want to prove that light is important for photosynthesis, then you skip the light part. Rather, and else you'll provide them with carbon dioxide, you'll provide them with water, you'll provide them with fertilizers. Only light is missing. That is your control group. Okay, a control group is that which would be given all the conditions required for an experiment except the independent variable. Okay, now what is independent variable? Independent variable is the one which you are testing for. If you are testing for light intensity, that would be your independent variable. If you are testing for flies, then flies would be your independent variable. Whatever you are if you are experimenting for temperature, let's say you want to check the effect of temperature on something, then temperature would be independent variable. Okay, got it? Okay, dependent is dependent on independent. Whatever changes because of independent. For example, light can affect the rate of photosynthesis. The rate of photosynthesis would be dependent. Okay, so I'm giving you example. Light on rate of photosynthesis. This would be independent and this would be dependent. Okay, got the difference? Okay, okay, there are two different things. One is control group and what is control variable. Okay, control group is the one which I've told you would be provided with everything except one thing. Now, control variable is the one which remain constant. For example, as I've given you the example earlier, that you want to check the effect of light only, then you would provide it with carbon dioxide, you would provide it with water, you would provide it with fertilizer or minerals. Okay, these things would be kept constant every time. Let's say if we are giving 100 ml of water, all the plants should be given 100 ml of water. If we want to maintain the temperature of 25 degrees Celsius, then throughout the experiment the temperature should be 25 degrees Celsius. Okay, whatever thing is constant is your controlled variable. Done? Okay, this is another thing in experiment. Okay, so let's move back to Reddy's experiment. Okay, um, so he said, what was his let's say the hypothesis, or what was his stance? He said that there must be something that is dropped by the flies on the meat that start the production or reproduction of maggots. Okay, later on the scientists found that there are microorganisms which actually reproduce and they form maggots. Okay, this is known as spontaneous theory of spontaneous generation. Let me write here. Like, life cannot come spontaneously, you need some external factor. Okay, theory of spontaneous generation. Okay, then the final stance was given by Louis Pasteur. Okay, Louis Pasteur is a French one. So I have told you that the scientists around the world, they did the experiment on something, on the same thing, to prove it right or wrong. So Louis Pasteur said that yes, microorganisms are involved in the production, or in the production of maggot, or they can give rise to something new. Okay, what did he do? What did he do? He he gave the concept of pasteurization. Okay, let me explain simple pasteurization. You boil the milk before use. That is pasteurization. Why do we boil the milk before use? Exactly. So that they cannot affect the quality of the milk when you drink it, or they cannot spoil it, let's say. Okay, so this process is known as pasteurization worldwide, because he is the one who did this experiment to prove his point correct. Okay, so he has taken an instrument. So you have that notes, you have the link, right? So do you want me to share a picture over here? Okay, I'll share. Okay, he took a flask, something like this. Pasture. I said Pasteur's experiment. Okay, and he took a broth in it. Okay, the broth has certain type of nutrients that can feed the microorganisms. Now what did he do? He boiled that broth. Now the purpose is to kill the microorganisms. Okay, so later on he observed, because the neck is open over here, the neck is open, that air entered and the broth would get rotten after few days. Okay, but if he covered that this one opening, then the broth is going to like stay over there for a longer time, it won't get rotten. So he proved that air must have some kind of microorganisms that enter the broth, and they start to reproduce over there by using the nutrients of the broth. Okay, broth with nutrients, air enters through mouth, might contain microorganisms that would grow or reproduce. Microorganisms grow and reproduce. So he observed that this broth, it turned milky because of the microorganism that grow over there. Okay, both experiments are clear to you? Okay, now this is known as biogenesis. Okay, the word is simple. Bio mean life and genesis mean the creation. The creation of life comes from the pre-existing cells. The living organisms, they arise from the pre-existing cells. So living organism only come from the living one, not the non-living one. This is biogenesis. Okay, life from life, let's say. Life from life. Non-living things cannot give rise to other organisms. Okay, done with this part. Now the next part is discovery of cell. We discussed this quite short, that there was a scientist who observed a honeycomb under the microscope, and he observed that there are compartments, honeycomb like compartments, and he named them cell. It was a simple thing. So the very first scientist was Robert Hooke. He observed honeycomb. Honeycomb mean the place where those bees live. Okay, and then he observed the cork. Okay, you know what is cork. Oh, cork, what is cork? A part of wood. Okay, he observed that under a light microscope. Since the microscope was not much developed at that time, so he observed that there are some compartment like things like this, and he named these compartments as cellulae, which means cell. So where were we? We were discussing the hook. Okay, he observed the cork and he observed that there are small compartments and he named them cell. Okay, there was another scientist who Leeuwenhoek. His basically his hobby is to make magnifying glass, testing magnifying glass. Just like kids, he roam around with magnifying glass. So he observed the pond water, and what he observed that there are small moving things in the pond water, and he named them as animalcule, which later on it observed by the other scientists that those are the microorganisms. Okay, but this was the name given by Leeuwenhoek, animalcule. Done. Okay. 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 Do okay, have you seen the movie Around the World in 80 Days? No. Okay, so there's a book on this as well, and in that movie they have mentioned the Royal Society of London. So there was, like, in the past, there was like an organization where scientists used to sit and where they put forward their theories, and the other scientists, they fight for, like, that their point is correct and others are wrong. So there was like a big, you can say, a company type thing, institute type thing, which is known as the Royal Society of London. So he published his research over there, and it was like worldwide known at that time. So anyone who want to publish something, who want to name something as his discovery, he approached that Royal Society of London. Okay, it's just an information. Okay, then there were another scientist known as Sheldon and Schwann. I hope I have written the correct spellings. I hope, yeah, it's correct. Sheldon, and the other scientist was Schwann. Okay, these are the one who gave us the postulate of cell theory that we discussed earlier. Okay, The cell theory is made by Sheldon and Swan after observing plants and animals. One observed plants and the other observed animals. Okay? Sheldon observes the animal and Swan was the one who observed like plants more. So they just combine their theories. Okay? Okay. Then next topic is microscope. Though we have done it in detail, let's have a summary of this. Sorry? Yes. Oh wait. Wait, I haven't seen that. Wait. Let me one more time confirm it. Yes, it's there. Microscope is over there. Stem cell is over there. Animal and plant cell is over there. Yes, it's there. Okay, let's see. Let's have like what we're discussing, the microscope. Okay, microscope is not that much stuff, it's easy. We use, there was a simple microscope earlier, then there was a development of compound microscope, which is light microscope, use light as a source to view objects. Light as a source to view objects, but there are few limitations. I guess I've shared the notes of these microscope with you. There are few limitations which are like they cannot observe the internal part of the cell, very first thing. Second, the life processes cannot be observed with the help of light microscope. Okay. 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 And if we talk about the general structure of light microscope, there is an eyepiece, and then there is an lens, and then there is objective lens. Let me give you a general idea of this. Okay, this one is known as eyepiece, which contains one type of lens, which is ocular lens. Sometimes it's 10 time, it makes the image 10 times more bigger, 20x, or it can be 40x, based on how much is the magnification. Okay. Then there is objective lens. Objective lenses also have different varieties. Some have 100 time magnification, some are like 200, some are 40. So the total magnification is made by both. For example, if you're using 10 over here and let's say 40 over here, so the total magnification would be 400. Okay. Okay. So the specimen is mostly placed on the stage. There's a like hole clippers given, and then light is passed through a condenser lens, which makes the specimen visible. Without light, the specimen is not going to be visualized. You need light to view the specimen. Okay. Simple. Done. Okay. The next thing is the electron microscope. We have done two types of electron microscope. One is the scanning tunneling, SEM, scanning electron microscope, and the other one is TEM, the tunneling or the transmission electron microscope. Okay, let's talk about the electron microscope. Okay, here light is not used, as the name indicates. It uses electron beam. It uses some kind of electric current, let's say, to view objects. Okay, its diagram would be like this. So there would be a source of electron, that is the electron gun. An electron beam would be passed through the specimen, and it would be viewed. It would be viewed over here. Okay. Okay. Okay, the benefit is that you can view even the tiniest thing, and you can observe the life processes. Okay. Okay. Then there is another type which is known as the laser scanning microscope. Laser, have I given you the types? No, I have written, I guess, or it's erased. Let me write again. Scanning and transmission electron microscope. The other one is laser electron microscope. Okay, why is this used? This is used to observe the thicker objects. Like some objects are so thick that they cannot be viewed under the microscope, under the electron microscope or the compound microscope. So what do? We put this under this electron microscope and it uses a laser. Name is indicating it uses a laser, and laser basically convert this one into a 3D structure. Convert the image into 3D structure. Okay, the source would be laser. Okay, okay. The power of okay, let me check if you need to. Okay, micrographs are very important. You need to like know the different diagrams, like how does different cell look like. Your next objective is that. Okay, now let's move to stem cells. We have done stem cells in detail, that there are certain type of cells in the body that gets differentiated into specialized cells. So when there is an egg, it's a simple egg. When it get fertilized, it turned into